On April 6, 2016, a baby boy was born in Mexico. That's not surprising. Babies are born every day all over the world, but this kid was special. A lot of us have two parents to thank for our existence, but this boy was co-authored by a whole team of scientists and had 3 genetic parents. And he's not the only one. Despite the ethical debates and complex lab techniques involved, more 3-parent babies have been born since then. There are some excellent reasons why you'd need a bonus genetic parent. But as I'll explain, there's still a lot we need to learn about the science underlying it, especially as those babies grow up. Let's start with the basics. To make a 2-parent baby, you need sperm and an egg. These 2 cells collide, fuse, and combine their DNA to make a full genome. Most of that DNA is nuclear DNA, which is contained in a cell's nucleus. It codes for things like eye color, hair color, and height, plus lots of invisible processes that keep your body running. But that isn't the only kind of DNA you have. You've probably heard of the mitochondria as the powerhouse of the cell. Cue flashbacks to high school biology. But powerhouses would be more accurate, since each of your cells contains hundreds to thousands of mitochondria. They help break down glucose and create ATP, which are energy packets that your cells use for everything, from muscle contractions to chemical reactions. But mitochondria also have their own unique DNA, called, you guessed it, mitochondrial DNA. Scientists aren't positive how mitochondria ended up with this genetic contraband, but one of the most popular hypotheses is that sometime over 1 billion years ago, a bacterial cell and a more complex host cell loved each other very, very much. So the bacterial cell moved into the host cell, helping it metabolize better, and eventually just became a part of it. Fast forward to today, and that love story left you with 37 extra genes, all related to cellular energy. So going back to that 2-parent baby, both the sperm and the egg come to the party with nuclear DNA and mitochondrial DNA. But soon after fertilization, the 100 or so paternal mitochondria are destroyed, meaning that only the mitochondria from the egg cell get passed to the baby. And it's probably a good thing the paternal mitochondria don't make the party's guest list, because sperm are super bad at proofreading their mitochondrial DNA, leading to boatloads of mutations. Mutations in DNA aren't inherently bad, but having more of them generally means that you have a higher chance of harmful mutations. So all the mitochondria and mitochondrial DNA a baby gets came from the egg, making it a gift from mom to you. But if that egg happens to have mitochondria with harmful DNA mutations, those mutations can lead to mitochondrial diseases, and that's not something any parent wants to give their child. To make matters more complicated, a parent might have a mix of healthy and mutated mitochondrial DNA throughout their cells, which is called heteroplasmy. And even if the parent has so few mutated mitochondria that they don't have disease symptoms, their eggs can tell a different story. Some of their eggs might have a ton of the mutated mitochondrial DNA, while others might have almost exclusively healthy mitochondria. It's really luck of the draw. Then there's the people who only have mutated mitochondria, which is called homoplasmy. And that pretty much guarantees that their eggs will have mutated mitochondrial DNA, and their offspring will develop a related mitochondrial disease. Unless, of course, you get a fertility lab involved in your baby making, and a 3rd parent. Using mitochondrial replacement techniques, or MRT, scientists can keep the original 2 parents' nuclear DNA, but swap in a donor's mitochondrial DNA. And that means all the obvious stuff we usually associate with genetic inheritance, like hair or eye color, is coming from the 2 parents who contributed nuclear DNA. And the donor mitochondrial DNA contributes to critical, behind-the-scenes things, like cellular energy production. There are different kinds of MRT being explored, but 2 techniques are the most popular. And just for simplicity, I'll use mom, dad, and donor to refer to the 3 sources of DNA involved in these procedures, though fertility clinics help all kinds of families. One method is the pronuclear transfer. For this, 2 eggs are fertilized by the dad's sperm. One egg is the mom's, and the other is the donor's. After fertilization, the nuclear material gets scooped out of both eggs, and the stuff from the mom's egg is put into the donor's. That creates an embryo that has the mom's nuclear DNA, but the donor's mitochondrial DNA. If everything looks a-okay, then the embryo can be transferred into a uterus to get growing. But that whole destroying a nucleus thing does mean that one fertilized egg is sort of destroyed in the process, and that can be a bit uncomfortable for some people. Another method is the maternal spindle transfer. Here, the eggs' nuclear material are swapped before fertilization. That creates an egg with the mom's spindle apparatus, which are the bits that move chromosomes around, and the donor's mitochondria, which is then fertilized with the dad's sperm. Then the same rules apply. If it passes inspection, it can be delivered to a uterus for growing. This was the method used for the 3-parent baby in 2016, since it avoids the ethical concerns around discarding an already fertilized human egg. So that covers the how of someone getting a bonus genetic parent. An extra parent, if you will. And while mitochondrial replacement techniques open the door for certain groups of 3 to have children genetically related to all of them, the main use so far has been to help 2-parent couples avoid passing down severe mitochondrial diseases. That was the case for that MRT baby born in Mexico in 2016. His chromosomal mother was a heteroplasmic carrier for something called Leigh syndrome. Leigh syndrome, also called Leigh's disease, is a heritable mitochondrial disease that causes nerve cell death and is usually fatal to children by 3 years old. Unfortunately, this mother had already lost 2 children to Leigh's disease, in addition to having 4 miscarriages. And when the researchers sampled mitochondrial DNA from her eggs, nearly 100% carried that mutation that causes Leigh syndrome. So MRT was pretty much her only option to avoid passing down the condition, while also maintaining a genetic relationship to her child. And the spindle transfer procedure led to the birth of a healthy baby boy. He was the first MRT baby, but he certainly wasn't the last. At the time we filmed this video, 8 MRT babies had been born in the UK after their mothers were confirmed to have pathogenic mitochondrial DNA and went through these fertility treatments. But mitochondrial diseases aren't the only reason scientists are working with families to create MRT babies. They're also researching more general causes of infertility. But before we get into the details of how that's done, here's a quick ad break.
This SciShow video is supported by Brilliant, the online learning platform designed to help you become a better thinker and problem solver. In school, you might have felt like your brain just isn't set up for math, but the people at Brilliant believe that everyone is a math person. It's the way we learn math that can feel confusing and hard. With Brilliant, you don't just memorize formulas, you get hands-on with concepts until they make sense. Brilliant helps you develop the intuition and problem-solving skills to figure out foundational math all the way to college-level calculus. To learn for free on Brilliant, go to brilliant.org/SciShow, scan the QR code on screen, or click on the link in the description. They're also giving you unlimited daily access to everything on Brilliant with 20% off an annual premium subscription.
Some researchers think that in cases where the cause of infertility isn't clear, the problem could be hiding in the cytoplasm of the egg. Since MRT plops one egg's nucleus into a donor egg, that fresh egg and cytoplasm might have what's needed to support pregnancy. To test that hypothesis, a 2023 paper published in the Journal of Fertility and Sterility investigated whether MRT using the spindle transfer method could benefit 25 couples who struggled with infertility in Greece. Over the course of the study, 19 3-parent embryos were transferred, and in the end, 6 babies were born. Maybe that means MRT can help with mystery infertility, but considering this was a small pilot study and 19 hopeful couples were still left childless, it's definitely not the only answer. Plus, that was one of several studies to reveal a major flaw in MRT. The chromosomal mother's mitochondria can be stubborn as heck. Because these embryologists are working with tiny single-egg cells, some mutated mitochondria can hitchhike into the donor's egg when the mom's nucleus is transferred, and then out-replicate the donor's healthy mitochondria over time. One of those 6 babies born to couples in Greece had less than 1% maternal mitochondrial DNA when it was a wee blastocyst, but that spiked up to around 30 to 50% at birth, based on samples of cells from different tissues. Scientists call that bounce-back reversion, and while it wasn't a huge concern for this infertility study since the parents all had healthy mitochondrial DNA, it's a major concern when MRT is being used to prevent mitochondrial diseases. Reversion even happened with that first MRT baby born in Mexico. At birth, some of his samples contained over 9% of his mom's mutated mitochondrial DNA. But luckily, it seems like 9% isn't enough for someone to get symptoms of Leigh syndrome. The mix of 2 parents' mitochondrial DNA in one cell has actually been a concern since experiments in the 1990s. Back then, scientists were considering whether a technique called cytoplasmic transfer might help with infertility. In cytoplasmic transfer, the cytoplasm and all the stuff inside, including mitochondria, from a healthy donor egg is added to the intended parent's egg. The idea was that some fresh cytoplasm might cancel out whatever was going wrong fertility-wise. These were technically the first 3-parent babies, but it seems like having a mix of 2 types of mitochondrial DNA might be dangerous, even if there aren't pathogenic mutations. We don't know the exact record of how many babies were born from this research, but we think it's about 30 or so. And we know that at least 2 or 3 of those children ended up with serious complications with their chromosomes or development. And because of those cases, the scientific community now seems to agree that cytoplasmic transfer isn't safe in humans. Plus, studies involving mice also suggest that even mixing 2 healthy mitochondrial DNA sources can cause cognitive impairment and behavioral abnormalities. Regulatory bodies across the world have struggled with how to balance the potential benefits of MRT against these kinds of safety concerns from tinkering with reproductive genetics in the lab. In 2015, the United Kingdom became the first country to explicitly legalize MRT while addressing the challenge of scientific oversight. They have a regulatory authority that grants clinics licenses to perform the procedure and reviews each potential patient on a case-by-case basis. And only one fertility clinic currently can use MRT. Essentially, the country decided that highly regulating the procedure was the best way forward. In stark contrast is the United States. After those cytoplasmic transfer safety concerns, the FDA decided in 2001 that any future egg experimentation would require their approval, and Congress also practically banned the clinical trials necessary for that FDA approval. So while MRT isn't technically banned in the US, there isn't a clear path forward either. The majority of countries around the world are taking a wait-and-see approach, like Singapore, or they haven't really considered regulating MRT beyond some murky laws, like Mexico and Ukraine. Because of this lack of standardization, it's hard to say exactly how many 3-parent babies have been born in total. We mentioned those 8 MRT births in the UK, and the fertility study in Greece that led to 6 babies. At least 7 babies have been born using mitochondrial replacement techniques in Ukraine, and let's not forget that first MRT baby who was born in Mexico. That is around 22 babies. And if you want to include the 30 or so that were conceived from cytoplasmic transfer back in the 90s, then you've got maybe 50 humans walking around with 3 people's DNA, but again, nobody's really tallying, aside from us, just now, with incomplete information. So, hard to know for sure. The science of making a 3-parent baby is still in its infancy, and even with the challenges of keeping track of these babies and the risk of reversion, scientists are hopeful about the future of MRT. At the time the paper came out in July of 2025, those 8 babies born in the UK were between 5 months and 2 years old. None of them had any major or permanent health concerns, even though one of them had around 16% mutated mitochondrial DNA at birth. That first ever MRT baby who was born in Mexico is only 9 years old now, so it'll be a while before scientists can accurately assess the long-term safety of these techniques. But since the future of MRT could involve addressing infertility, helping non-traditional families grow, or avoiding fatal mitochondrial diseases, it's probably worth the wait.